Hi, my name is Gregory Block. I'm the SVP of Corporate Development in Notch Therapeutics. Today, I'm going to tell you about what we're doing at Notch Therapeutics to advance pluripotent stem cell-derived T-cell therapeutics. Some highlights about Notch. The company was founded in 2018. We licensed technology from the University of Toronto and Sunnybrook Research Institute from the laboratories of Juan Carlos Zúñiga-Flucker and Peter Zandstra. In 2019, we partnered with Allogene, which I'll talk a little bit later in the presentation. And in 2021, we closed an $85 million Series A investment round. We are growing rapidly. We currently have now over 60 FTE in Toronto, Vancouver, and Seattle. One of the core philosophies of the company is that we licensed this IP, but we also married it with these very core pillars of capabilities, including manufacturing and advanced cell manufacturing, product development, and research and bioengineering. And I'll touch upon all of these in my presentation today. So as I've mentioned, we are focused on pluripotent stem cell-based T-cell therapeutics. We believe that pluripotent stem cells can create a truly allogeneic product because of their capacity to divide and be manufactured and quality controlled. So when we compare these to the autologous or donor-derived solutions that are out there, we can reduce variability, we can increase homogeneity, we can reduce cost of goods, and we can improve accessibility of these medicines ultimately. The core of the technology we licensed is called the engineered thymic niche, or ETN. And what the ETN allows us to do is maintain maximum control over the delivery and interpretation of Notch signals that T cells get during their development. So as some of you may know, Notch signaling is obligatory. Activation of Notch signals is obligatory for T cell development as they arise during hematopoiesis. So what we do as part of this engineering thymic niche is we coat these microbeads in the right concentrations of delta ligand, which is a Notch ligand, and an accessory factor called VCAM1, which enhances the signal delivery of Notch to the developing T cells. By putting these signals onto microbeads, we have full control over the ligand concentration, the timing parameters. We can control all these parameters independently, and we can coax out different fates of these cells as they go through hematopoiesis in a dish. The delivery of Notch signals to developing T cells is not a new concept, but there are major advantages to the ETN platform that are really pertinent to manufacturing of a T cell product. So in this schematic above, what I'm showing is a starting material, in this case, a gene-edited pluripotent stem cell. So we have a gene editing group where we can introduce our desired edits into that pluripotent stem cell that then make its way into the final product. We differentiate those cells towards the CD34 hematopoietic progenitor cell fate. These can then be staged again at the progenitor T cell fate, which are the canonical markers there are CD7 and CD5. And then those progenitor T cells can go on to develop mature functional single positive CD8 T cells, which will recognize, tumor-associated antigens on cells in a dish. The technologies that have been used in the past or tried are listed here in the bottom table. Soluble DL4, to stimulate Notch signaling alone does not really cut it. Although it doesn't include any animal components, you don't really develop T cells by doing this. You mainly skew the cells towards a B cell fate. The original technology to enable the production of T cells from hematopoietic progenitor cells was published in the 2000s by Juan Carlos Zúñiga-Flucker, our scientific co-founder, who showed that if you express these delta 4 ligands and delta 1 ligands on the OP9 feeder cell line, that you could stimulate T cell production from these progenitor cells. Other groups have tried artificial thymic organoids that allow the T cells to go through their positive and negative selection and fate determination in little thymic organoids in a dish, which is a fairly complex system for expanding T cells for clinical use. Whereas the notch technology allows us to scale either in a two-dimensional format or on beads in a three-dimensional format to achieve scale without the use of any xenogenic compounds or other cell types. So you're not, when you're manufacturing your T cell products, you just have to worry about this one ancillary reagent, which is fairly straightforward to control. One of our cornerstone functions in Notch is our computational biology team, in which we use computational biology to predict development behavior of cells. So we can go in and query what's happening to different signaling pathways at what time. And that allows us to generate iterative hypotheses based on fundamental data of the underlying biology of the cells. And that allows us to solve a number of challenges in the organization. One is understanding the way the cells are going through development in a dish, but also ultimately on what parameters will allow us to predict efficacy of the cells. How will we model PK and PD parameters for the products that we develop? And one of the outputs of these capabilities that I've just described here is our ability to sort of peer into what's happening in a notch signaling pathway during development of these iPS-derived T cells. And you can see here on the left-hand side, what I've got is a couple of heat maps that are showing the relationship between the different notch signaling genes. And you can see that as we increase the amount of notch ligand that's presented to the cells on our beads in collaboration with VCAM1, we're able to activate notch signaling throughout. So we're not just looking here at just one marker, we're getting a whole view of how notch signaling is feeling in our cells right now. And you can see by increasing the amount of delta ligand and VCAM1 that the result of that activation of notch signaling is an increase in the CD5, CD7 pro-T progenitor cells that will ultimately give rise to CD8 positive T cells during our production protocols. I'm going to pivot now to talk a little bit more about the company. As I mentioned in the first slide, we have 3 sites in Canada and the USA. We have our headquarters here in Vancouver, British Columbia, and our R&D center. So here is where we do all of our particle development for the engineered thymic niche, production of the engineered thymic niche. Our systems biology team and our computational teams are located here. Synthetic biology design, our differentiation and PSC, stem cell developmental immunology is all located in Vancouver. We collaborate very closely with the site in Toronto, where we have our process development groups and our GMP manufacturing groups, where we're creating, what I think are the best GMP lines in the world, and pluripotent stem cell lines that can give rise to different cell products. We're scaling up in PD. We're bringing things into suspension bioreactors. We're doing clinical grade gene editing, assay validation, all to be ready for taking these cells to the clinic to bring value for patients. And we've recently opened offices in Seattle as well. This gives us a US footprint, and we've built a preclinical and translational sciences team led by Chris Bond, who I'll introduce you to on a coming slide. So we've put together quite an outstanding leadership team at Notch. David Main has joined us as CEO. He was the previous CEO of Equinox Therapeutics that led a successful reverse merger with Neoleukin Therapeutics. Joining him was Cam Alam, our CFO, who led business development activities and finance activities at Equinox and Neoleukin as well. As I mentioned, Peter Zanstra is our Chief Scientific Officer. He's the founding director of the UBC School of Biomedical Engineering, and has a lot of experience founding massive institutes for stem cell biology and stem cell biomedical engineering. The middle person is me. You're looking at me. I'm wearing a white shirt, not a yellow one today, so it's a little bit different. Christopher Bond leads our preclinical and translational sciences team in Seattle. He joins us from Juno and Kite, where he has enormous expertise in developing T cell products derived from pluripotent stem cells. Emily Titus is our VP of Process Sciences, who leads all of our GMP activities. She was previously at CCRM, where she helped build out their clinical gene editing strategies and capabilities. And Bernie Lamb also joins us to lead finance from Aquinox and Neolucan. We have a scientific advisory board that's led by our scientific co-founder, Juan Carlos Zúñiga-Flucker. He's joined by Chris Sturgeon, Yvonne Chen, Jeff Hubble, and Marcel Vanderbrink. So we have lots of very interesting conversations about the directions we can take our science and discuss in depth our scientific challenges that we need to solve moving forward. As I mentioned, we have some very important collaborations to drive these products forward. The primary collaboration is with Allogene Therapeutics. This deal was done in 2019 and brought a lot of enabling capabilities and capital to Notch through an upfront payment, an equity investment, R&D investment. We're working very closely with the Allogene team to bring named CAR targets with primary applications in heme malignancies. Notch is eligible to receive milestone payments per exclusive target, and ultimately could enjoy mid to high single-digit royalties on net sales of these products. CCRM as well, we work very closely with this team in Toronto for our GMP manufacturing and gene editing. They are a founding entity of the company and are a very close partner for helping us achieve our lofty goals for creating these clinical grade products. Looking forward, we are shaping out a pipeline of products based on pluripotent stem cell derived T cells. We have our partnership with Allogene, which is driving our direction for hematological malignancies, while we're focused primarily on solid and hematological malignancies within the company, developing our own proprietary differentiated products in this space. We also have research programs and collaborations looking at other types of cells that could be derived using our technology. For example, T regulatory cells, which could be used in autoimmune diseases, and other immune cell types as well are of interest to the company. And we, of course, are open to discussing any potential partnerships in the community. So please reach out to us if you have an interest. And just a summary slide of our value proposition. We believe that pluripotent stem cell based T cells will help with a lot of challenges being faced by the sort of first generations of donor derived cells. We're able to check a lot of boxes under the clinical, regulatory, and commercial buckets that one would think about when we need to create a product like this. For example, I'm not going to read the list here, but for example, having one cell that you can create a master cell bank of, characterized properly, making sure it has all the right reagents used so that you're able to take this cell that you've invested in early all the way through to commercialization is critically important. And we keep this top of our minds as we're going through even these early phases of development. And ultimately, that'll allow us to lean much more rapidly into commercialization. We'll be able to consolidate our manufacturing footprint, have feasible distribution, achieve a large scale that you need to meet patient demand in the future. With that, I'd like to thank the organizers for inviting us to speak today at the conference. Again, we're so sad that we can't be there. I missed the courtyard at the nice hotel there. But I hope you're all having fun and that you have a drink on behalf of Notch. Thank you very much.